Name any organ and you can find a cell undergoing mitosis. Meiosis, on the other hand, is specialized for the reproductive organs. We've got a nice little germ here. No, not a bacterial germ, a cell germ. This diploid cell is going to split into 4 haploid gametes in order to fuse with another in fertilization. In this cell, we can see that DNA is still in its uncondensed strands forms. As the chromosomes are condensing, the nuclear membrane begins to dissolve, making it easier for this material to move around. Here's the diploid cell before replication. There are 2 homologous pairs depicted here, soon to be 1 for simplicity. One color depicts one of each chromatid from each genetic parent. Now begins the S phase, where the pairs duplicate, bound together by a centromere. These doubles are called sister chromatids. It's important note to not confuse them with a homologous pair, 2 different pairs of sister chromatids. Remember how I mentioned each color has a different parental origin? These pairs may be different, but they code for the same proteins. Genetic diversity plays a massive role in a population's survival. In order to spice things up, these chromatids swap the same chunk of DNA with each other. This process of crossing over mixes up the alleles being passed down to offspring. We'll come back to this. The cell's 2 centrioles, the little churro-like things, assist the spindle fibers in grabbing each one of the chromatids. They then move to opposite sides of the cell, where they are pulled apart from the centromere. The cell begins cytokinesis. In this cell, a cleavage burrow appears, lysing and splitting the cell. It is important to note that at this point, organelles like mitochondria are distributed between the 2 sister cells too. Mitosis 2 begins and the little centrioles duplicate, repeating the pull behavior from meiosis 1. Chromatids are pulled apart, only 1 to each side of the cell now, and the burrow begins again. Each of the original sister cells are split, and for new and final haploid cells, 1 chromatid for each different chromosome remain. What used to be originally 1 big diploid cell has transformed into 4 smaller haploid cells. These cells are called gametes. Now I said we would briefly come back to crossing over, and looking at these 4 cells, it is apparent that each chromatid is distinctly different from the other 3. This assures that offspring will have sufficient genetic diversity for survival. That is it in meiosis. Sorry, fake fade out. We'll brush up briefly on fertilization. Gametes like shown will become an egg or a sperm, depending on the sex of the individual. In a case of fertilization, 2 of these haploid cells will fuse together, becoming 1 diploid cell, just like all the other cells we see in our body. This zygote must grow and grow via mitosis. Fetal development is not a part of this lesson. Thanks for sticking by. Bye!